Hello and welcome back to this lecture 21 of biomems. We'll do a quick preview of what was done in the last class. So we talked about the self assembly of nanoparticles and structures using DNA molecules, wherein basically the complementarity of one strand with another was used as a property to assemble one or more nanoparticles. The strategy could be used for making interconnects at the nano level, wires, etc. So we also talked about DNA hybridization as a strategy to detection and discussed microarrays. Essentially they are libraries of different capture probes which can be used to hybridize the DNA specimen, the target DNA molecule, and then you can actually label the target with the fluorophore, so that as it goes on to the capture probe and gets immobilized onto the surface, you could do a fluorescence readout and see whether there is a binding kinetics which is happening between the capture probe and the target DNA. So we talked about a little bit of concepts about microarrays, features, what exactly hybridization is or how it can be done or how sensing can be done. And then we also discussed about the various utility aspects of such microarrays, essentially RNA transcription profiling or HIV resequencing. All these different things can be studied using DNA microarrays. Okay. We also talked about two different companies and two different approaches that exist in the market as on date for these different DNA microarrays. The two companies are Affymetrix and Nanogen. One of them, actually the Affymetrix, is one which does a light directed synthesis of these capture probes. So essentially by using several photomasking strategies and chemical steps in between, so there is a buildup of molecule by molecule. So you can have several combinations of these different ATCGs in order to realize a variety of capture probes in different points of space, okay, over the whole array. And so therefore you could actually open and close pixels by doing multiple steps, so that you could build exactly the molecule that you are looking at and have an information of the sequence of the capture probe. Nanogen, on the other hand, is a company which actually gives you the chips and lets you assemble the capture probes that you want. Okay. So you have to buy the capture probes separately, and this is also known as the electronic way of immobilizing capture probes. Essentially there are a bunch of electrode arrays on a surface, and you are using a positive potential to pull selectively on some few posts, gold posts, these molecules, these negatively charged DNA molecules, which would thereby go and bind on the surface. There is a binding film like poly and lysine, which is essentially positive charge, and there is electrostatic attraction between the negatively charged DNA and the surface. So you have a build up different over these different electrodes, all different kind of arrays, and you could actually use a negative potential on the others, so that you could direct only to the particular position in which you want the capture probe. And so it has a very specific process for doing this immobilization of capture probe. So we talked about these different approaches, and then also discussed about the DNHF from Motorola, which uses electrochemical sensing using a FECN label and a bunch of different molecular linkers and wires as standing up on a surface upright as a brush polymer, and using of signaling and a capture probe in order to capture a certain target and position the FECN 6 or the the ferrocyanide probe very close to one of those molecular wires to take up the current and then read out what is there as sequence on the target DNA. Okay. So today we will actually start with a new area which is also known as the fundamental process for gene sequencing. It was developed way back in 1975 by a scientist named William Sanger, and this process has been popularly known in his name as Sanger's reaction. So this is again a very interesting and fundamental level technique for identifying the sequence on the genes. And by sequence what I mean is, suppose there is a DNA molecule as shown here, and reading out from the 5' to 3' you can have these different groups A, T, C, T, G, this again G, C, and so there is a sequence of these molecules. So essentially the sequence is a very important piece of information because it gives an address of the particular biological moiety or entity which contains this. Okay, it may be unique to a certain bacterial cell or a certain virus, and it may be able to identify if the virus is there in the ambient just by looking at the sequence of the DNA that it has. So it's a very important information which is now, after the Human Genome Project, being recorded in a large database which is maintained at a global level, and research gets on added where these sequences are identified for any new stain or any new bacteria or any new virus, and it's essentially put up there. It's also known as the NIH database. Okay. Now how do we identify the sequence? This very famous Sanger's reaction is all based around this molecule which is a dideoxynucleotide. Okay, so you can see the schematic of the molecule here. It's a dideoxynucleotide, so therefore there would be a dideoxy chain terminated A adenine, dideoxy chain terminated cytosine, guanine and thymine respectively. So there are 4 different dideoxy chain terminated molecules. Okay, now the way that this goes is that there are the several different constituents in the Sanger's reaction. There is an enzyme, this DNA polymerase, which essentially synthesizes the broken strands of the DNA. There are 4 different DNTPs, which is the normal A, T, C and G. It is not the dideoxy chain terminated NTP. And then we have 4 different reactions. So this process is carried out in 4 different steps. Okay, in each of these steps there is a different dideoxy ATP, dideoxy CTP, dideoxy GTP and dideoxy ATP essentially. Okay, so let me just write all this down here for your convenience. So you have reaction 1 with DD-ATP, number 2 with DD-CTP, 3 with DD-GTP and 4 with DD-thymine nucleotide. Okay, the dideoxy chain essentially is an end group which stops the reaction. So whenever you have the addition of a dideoxy chain, it doesn't let the DNA chain replicate anymore. It essentially stops the replication process. So the chain is broken at that instance. So suppose if I have a reaction wherein in one go the chain is broken along A essentially, so I can actually have a DNA molecule now from the 3' end all the way to about A. Okay, if the reaction goes all the way up to, let's say, this particular G here, so I can now have the in another reaction the chain terminated from G. So therefore 3' to G will be there on the chain. So whenever you put these different dideoxy chain terminated agents, which is also about 1% by volume of the NTPs, into the reaction and then independently do the 4 reactions. So wherever there is a thymine and this gets bonded to a DD-ATP, it automatically cleaves the reaction. So there can be a fragment wherever there is a thymine, there is a fragment of that particular length. Okay, so if you put if you try to put this DD-GTP on the reaction, so wherever there is a cytosine on the principal DNA strand, the DNA will break, and so there will be multiple lengths of DNA available as an end product of all these solutions, okay, or all these reactions. But mind you, these are terminated only at a single group. Like for example, if it is a DD-GTP, then it will terminate only at the cytosine groups in the DNA. If it is a DD-TTP, then it will terminate only onto the adenine groups on the principal DNA. So once these products are run through the gel, they have all varied lengths, and you can easily fractionate them using gel electrophoresis. So the smaller the DNA is, the faster it moves, the longer it is, the slower it moves, and essentially you will have a readout like this. So this is the DD-ATP. So essentially the chain termination here takes place along the thymine group. So wherever there is a thymine on the DNA molecules, you have fragments made of that level. So essentially these thymine terminated processes are represented as gels, let us say here, here, here and here, right. So, it gives an idea of the position or the length at which this particular group is present. So, wherever there is a TTP, dideoxy TTP, it terminates the molecule wherever there is an adenine, and so therefore, if you run the products, you have these different lengths of products which are coming out where there was actually an adenine on the parent DNA group. Similarly, in the DDCTP, you will have products which are kind of fractionated in a manner corresponding to wherever there is a G or a guanine, and similarly DDGTP, the products terminated in a manner wherever there is a cytosine in this particular group. So, you have these different instances, okay, where you can find out that how or where exactly what group is located. So, if you read this out as a whole, if you read this whole gel image and then try to see whatever comes and corresponding to that what it is really. So, if there is a TTP in the reaction, there really should be an A here, okay, and A here, there should be a T here, similarly there is a T here. So, essentially this kind of gives you a feel of what exactly it is that you are looking at, okay, and of the DNA. And if you just put this sequence all together, something like this, you get the total sequence on the DNA. So, this is essentially the process, right. In a nutshell, if you would like to summarize what happens. So, dideoxynucleotide is a kind of stopping mechanism for the extension, any further extension of the DNA strand, leading to the cutting the replication reaction at a certain point where the dideoxy group is picked up. So, if there are certain points which are corresponding to the complementary pair as that on the dideoxy end group, it is picked up and the reaction stops there. Similarly, the DNTPs with 1% DDNTPs are normally used for this process, and every time the product is run through a sieving gel, you have all different sizes which comes up on the gel from which you can read out really what is the sequence of the DNA, and essentially the readout process is also electrophoresis best in this case. Now, when we are talking about electrophoresis, I would like to just kind of reiterate again that there are different types of sieving matrices based on which you could have almost close to a 1 base pair resolution. For example, the different products like let us say pluronic, it is a trico block polymer, polyethylene oxide, polypropylene oxide, polyethylene oxide. This has essentially very small pores, and it is an absolutely transparent gel material. Within capillaries if filled, it results in a reusable or a washable capillary, essentially because this material goes into liquid or gel depending on the ambient temperature. There is an agarose material which is most commonly used in laboratories for this work of electrophoresis. There are polyacrylamide gels, there are products associated with hydroxyalkyl cellulose and a lot of different polymers of the kind which would have essentially a porous content and would also have some kind of storage for the buffer to be there in the channels, etc. So, these matrices are very often used to get these kind of readouts as I illustrated here just about a minute back, and from that you could actually read what is there on this particular DNA. Okay. Now, there seems to be a little problem though, and the problem is the speed, the rapidity which you can read this out. And so therefore, there have been initiatives or efforts in this biomems or diagnostics area where people have really tried to evaluate that can all this mechanism of this reaction and gel and readout so on and so forth be made made sensitive enough or rapid enough so that you could go to a very low time of detection level. Okay. So, for that there is a group in University of California at Irvine who are working on these strategies based on the atomic force microscope. Essentially the idea is to be able to pick up molecules on a very small minuscule level and use surface electrophoresis to shift the molecules back and forth in this particular manner by applying a potential. And then once these are all picked up using a hybridization array or something where there is a primary level detection, you could sequence it by running it on these small wells here and running the Sanger's reaction, picking up the products and then the idea is that as a label is kind of inserted into the particular sequenced product, you can see the fluorescence once it is moved down with a pulsating electric field. So as it crosses this self luminous tip here, it kind of gives out a fluorescence response from which you can back calculate what was there on the end, on the group on the DNA essentially. So you are at a stage picking up the correct DNA by using hybridization, and then picking it up using an AFM probe using a small voltage at the tip of the probe, and then essentially you are trying to drop this back down into small numbers into different wells, which would do these 4 reaction steps associated with Sanger's, and then the products, when they are all different lengths and they are kind of labeled with a certain fluorophore which is also corresponding to the end group which is there on the dideoxy chain, okay, or which is terminated at by the dideoxy chain, that particular molecule would come out at a certain rate, okay, where it will come out at a certain point of time from the AFM tip. The way you read the fluorescence, again, you create a small self luminous spot here, and then using an AC signal you can actually send this down, this DNA down. You have picked up the DNA here in this case from this particular well, if you can see, right, and so you have to send these different lengths down along the tip of an AFM so that it kind of goes one by one, and as it crosses the luminous point you get a certain fluorescence readout from which you can find out what was the base pair which was chain terminated, okay, or which had the dideoxy end group, and from that you could also find out what is the sequence in the DNA that you are picking up. So this is essentially a very interesting technique that people have been developing. I would also like to illustrate a little bit about what surface electrophoresis would be actually able to do in this particular case. As you are seeing, the first question which comes to mind is that why is it that people would do electrophoresis on a surface when there are already existing gels and other materials which would do this electrophoresis beautifully. One of the reasons why electrophoresis is taken from a gel or a volume into a surface is essentially because if you go above a certain base pair, let us say about 10 kilo base pairs or so, the gel materials, irrespective of how big the pores are, has a tendency of cutting down the resolution of DNA. So beyond a certain point, let us say if you are actually going above 10 kilo base pairs and trying to translate or fractionate DNA across this gel material, there is a model called biased repetition which comes into picture, which means that let us suppose the DNA is like a serpent which is kind of unfolded. If you may remember, we discussed this at length when we were talking about electrophoresis. So the head of the serpent goes into a series of pores, but then the DNA realizes it's too long to go through those bunch of pores into the next section, okay, as a result of which it decides to kind of stay back there and gets jumbled, so it does not move forward anymore. So this is a effect because of excessive friction between the walls of the capillary or walls of the pore through which the DNA is passing. To avoid this problem, people or scientists have investigated this process using an alternative strategy called surface electrophoresis, okay. So this approach essentially uses electrostatic interactions between the molecules and the charged surfaces for size fractionation. I will just about explain this diagrammatically in a little bit, and this technique of DNA fractionation gives an alternative to all these techniques in context of separation of higher base pair sizes. So it is a very good alternative that the surfaces have to provide. Okay, so how the surface electrophoresis is done, and this is essentially out of research work done at Benjamin Chu's group up at Stony Brook. So essentially if you look at that, when a DNA molecule is adsorbed on a flat surface, there is really a loss of entropy due to the restrainment of DNA molecules on the surface, okay. So there is also an increase in energy due to adsorption phenomenon, and there is a balance of these two energies which promulgates the DNA segments to be present either as loops or trains, okay. So you are having a flat surface and you are putting this or physisorbing this DNA on the surface, and you essentially have a layer of water over the surface, and the DNA is still adsorbed, physically adsorbed on the surface over which the water is present. So the DNA has a tendency to kind of balance the adsorption energy over the local increase in entropy because of the extreme amount of negatively charged backbone, and there is a configuration which comes out of it which is essentially like a loops and trains. So the portions of the DNA are looping into the solution as if they are kind of hanging out, and the portions of DNA are essentially adsorbed onto the surface of the solution. Okay. So people have shown with molecular dynamics very interesting images and results which can call for it. Let us suppose if you look at this particular figure here, you can see how using a ball and string model this person here has been able to show that there is really a tendency of this DNA to kind of get folded into a loop as well as these trains. Okay. So there are these trains here and there are these loops which essentially formulate. Now one interesting factor here is that longer the DNA molecule is, the more would be the train part or more would be the adsorbed part, and one of the reasons why that is so is because the loops essentially are a bunch of different negative charges. So if the molecule is very long, you could consider that the charges are too much to be in the solution. So there is a dense network of charge like this on a surface, right, and so if this DNA is long enough, there is a huge repulsion if it were to be present as loops. So it is definitely not a favorable energy configuration. So the other configuration that would be available would happen when they are really adsorbed very close to the surface and they are small. So the loops are basically small and the train part or the adsorption part is more. So we get one fact from all this, that the DNA molecule, if it is a longer molecule or it is a long molecule, there is a tendency of the train part to be more as opposed to the loop part, which closes or closely adheres it or adsorbs it on the surface. In this condition, if you try to apply an electric field and try to move it along the direction of the electric field, there is a tendency of the DNA to move slower by virtue of the adsorption energy which comes into play every time you have to go to the next step, or the DNA has to go to the next step, as opposed to the shorter DNA segment, where probably because of less interaction or less entropy and longer loops and less adsorbed area, the DNA moves faster. So this is actually a basis of size based fractionation of DNA, but just using surface without using bulk properties, and therefore all those other impacts which would have otherwise come from a 3-dimensional gel kind of matrix, where there are a bunch of different pores through which this DNA is moving and there is a lot of friction between the DNA molecule and the pores, you almost always get a bundling and a loss of resolution at higher base pairs. However, in the surface case, the surface and the environment around it automatically adjust the DNA to have smaller loops or longer loops, and essentially it just makes it move slower or faster, and there is a huge amount of resolution aspect in this particular case. So I would like to illustrate some of the work that has been already done. So this is the case of shorter molecules. Okay. So this essentially is the case of shorter molecules, whereas these are the longer ones. So you have more trains than loops. So in summarily, so whenever there is a DNA molecule adsorbed on flat surface, there is a loss of entropy due to restrainment of DNA molecule on the surface. There is also an increase in energy due to adsorption, and the balance of these two energies promulgates the DNA segment to be present as loops or trains. Okay. Yeah, if the surface is immersed in solution, then the loops extend into the solution, and the trains, which are contiguous segments adsorbed over the surface, are held close to the surface. The shorter the DNA molecules are, they have less negative charge on them, and so the repulsion force is not much, and therefore they form more number of loops extending into the solution than longer DNA molecules, which behave otherwise due to high coulombic repulsion. Okay, and they adsorb more on the surface rather than going into as loops. So therefore the smaller ones should move faster because they are less adsorbed on the surface. The only difference in this case is when that only higher size fragments electrophoresis better in this case. Typically it is 2 kpp onwards, but essentially the effect can be felt more from 10 kpp, because just like agarose could easily go up to 10 kpp. Of course there would be a resolution loss, but then still you could make it detectable up to about 10 kpp. So the utility of this process is felt beyond that, although they can actually start starting from about 2 kilobase pair size of the DNA molecule. So this is an excerpt essentially taken from Benjamin Chu's group, where they are talking about using this technique to detect the relative migration time of different sizes. So here you can see you have DNA of 2 kbp all the way to about 8 kbp, 8 kilo base pairs, and you can see that if you see the log of mobility, it kind of decreases as you increase the number of sizes. This is a plot between log of mobility of the DNA versus the log of number of base pairs, right. So if the number goes up here, as you see, the mobility goes down, which is in perfect agreement of what our theory is really. You have illustrated this from this particular plot here of the migration time versus fluorescence. Okay. So one more interesting factor which Chu's group has shown is that you can really detect with an AFM the DNA adsorbed on an OTS covered silicon layer after an electric field of 4.5 volts per centimeter is applied. Okay. So here essentially these arrows as you are seeing kind of indicate the shorter chains and the way they are moving. So the shorter chains are all moving together. If you see, these arrows kind of signify them. These are the shorter chains. Here essentially these are the shorter chains, and the longer chains kind of move separately, which is probably not shown in this particular figure, but Benjamin Chu's group has observed this essentially. Okay, so essentially a very repeatable kind of data on a surface of DNA has been recorded and repeated. And the fluorescence intensity spectra as a function of time for a 1 kilobase pair ladder on a bare SI surface is given in this particular graph as we discussed before. Okay, so I'd like to show you how some of the detection can be done, and this is in fact a paper by Kuo et al. Up at University of Tokyo. And so what he talks about is that you have a microfabricated architecture here. You are talking about an SU8 based microchannel with some reservoirs, and this is mounted in the top of 2 electrodes which are made up of chromium, okay, and essentially on a glass plate, and you connect these electrodes through an external battery, an external source as indicated here, and you basically run the charged DNA from the feed reservoir side, which is this, up to the detection reservoir side, which is this. So here of course they have shown using UV light at 254 nanometers the labeled DNA, the fluorescently lighted DNA with ETBr, etherium bromide, and this is of course a lambda DNA that kind of moves all through. So you can see these figures of the reservoirs, okay, and they can see that with time this fluorescence spreads from this reservoir all the way up to the detection reservoir. What they also report is an impedance based signal connected from the electrode at the detection reservoir end, where they see peaks coming as these DNA molecules would kind of go through and pass or hit the particular electrode, a positive electrode made up of this chromium. Okay, so this kind of gives or endorses this concept of surface electrophoresis. So our group has done some work on electrophoresis as well, and this is a most recent excerpt from one of the works reported in the American Institute of Chemical Engineers 2009 meeting. So essentially this talks about a surface, a silicon surface with a potential across it, wherein you are actually physisorbing some DNA molecules on one side of the surface. You have a PDMS upper layer which is acting as a cover and also gives a microchannel in between through which the DNA should be translated, and you have actually pasted the electrodes, film electrodes on both sides so that you can apply a strong field from where the DNA is adsorbed this side all the way to the other side here, and then you have connected these through external wires. Okay, and this whole assay is placed inside a very small flow cell, as you are seeing here in this. Number 1 is the flow cell, and you are filling this up or covering this whole thing up with the buffer solution. So the DNA kind of loops into the solution and forms the exact same configuration as was reported by Chu's group. Okay, so it is essentially brought under an inverted fluorescence microscope, as you can see here, which kind of gives us a readout, and these show some of the illustrations of how a DNA ladder would behave on doing surface electrophoresis. So this is a dye doped DNA. Similarly, this is after a few minutes of electrophoresis, surface electrophoresis has occurred. So you can see there is a marked difference between the way that the ladder looks before electrophoresis as opposed to after electrophoresis. This is simply physisorbed onto the silicon surface. I have some other data to share with you. Here is again the translation of DNA molecules across the surface at different instances of time. You could see all from 0 minutes all the way to about 23 minutes, and you could see that because you have applied a positive and negative potential in the following manner, you are seeing that there is a translation of DNA towards the positive potential. You can actually do through image analysis a scan on the overall boundary and try to detect the centroid and then calculate mobility. This right here is an illustration where we are talking about the mobility of such DNA molecules, and we find out that although the electric field is applied only parallel to the y axis, there is almost always a Brownian motion along the x axis which causes the DNA to move hither thither as opposed to going just straight away in the direction of the electric field. Okay, so the mobility of a 1 kilobase DNA on silicon dioxide surface with a voltage of 24 volts has been plotted here in this graph, and this is corresponding to a separation distance of the electrode of around 1 centimeter or so. So this is a perfect bidirectional process. As you can see in this particular illustration, what we did here is really we just went ahead and changed the direction of the electric field. Here this is negative and this is positive, and you can see slowly that the DNA kind of emerges back into the field of view for these different time intervals, as can be illustrated from 0 minutes all the way to about 10.03 minutes, and then we kind of plot the mobility on the same mechanism as we did before and still see the Brownian motion. Okay, so this is a real problem because you can't get a one dimensional mobility model, because of which we are trying other alternatives like kind of super focusing the DNA onto a very small track so that we can eliminate totally the Brownian motion. The super focusing can be obtained by using a different surface energy pattern on a surface where we can really make a narrow hydrophilic pattern where all the DNA would rapidly converge, and we are not sharing that data at this time, but I just wanted to make an illustration of what kind of research is possible in this particular area of surface electrophoresis. Okay, so this is some mobility studies that we did where we can show that on an energy on a higher energy surface like silicon dioxide as opposed to a lower energy surface like porous structure, porous matrix made up of silica, we have differential mobilities which we report, and we are trying to understand the process better, but essentially this is how surface electrophoresis can be done. So I now would like to illustrate another very novel technique of biomems which biomems has to offer based on the various fabrication protocols or various kind of structures and features that can be fabricated using microsystems fabrication technology. Okay, so this is really the diagnostics at micro nano scale, and is also the basic principle behind Coulter counters. So we are talking about some kind of a cell counter by the name of Coulter counter. So the whole goal here is the following. So you have a pore, a small pore which is close to the size of a single cell, okay, and the spore or the porous membrane is kept as the only communicating medium between two fluid cells, where one side is rich in the cells and another side is essentially a lean mixture. And if assuming that the cells are negatively charged and you apply a positive bias on the other side of the fence or the other side of the pore, there would be a tendency of the cells to go into the pore and go straight out into the other side, okay, and while doing that it kind of momentarily blocks the pore and blocks the ion current which flows in the reverse direction as opposed to the direction of the movement of cells. So this is simply based on this principle, okay. So you have a negatively charged cell here, and you have a pore, a micropore in silicon which is made like this, okay. This is the trans side or the trans chamber, this is the cis side, and you are applying a potential across this particular porous membrane in a manner that this negative cell is dragged into the trans chamber side. Okay. So, the idea is that you keep on monitoring the ion current, that means the current I with respect to the time t across this membrane, and the moment the cell goes and blocks the membrane, there is a dip in the ion current, and when it goes to the other side, as you are seeing here, the current kind of takes up back. So, if I can count these dips with time, I can be able to ascertain how many cells pass per time from this particular micropore, and this could be a basis of counting the number of cells that are there in the rich side of the solution. Okay, so this is the basic principle of micro nanoscale Coulter counters. Based on that, based on this micro scale Coulter counter, you can extend the same concept onto the nanoscale. In fact, the first one of the first few articles which came out in this area was really from Kasyanovitz group, and would like to illustrate one of that example here, where they talk about self assembly of a protein by the name of alpha hemolysine on the top of a lipid bilayer of a cell. So, if you see here, this is essentially the lipid bilayer that they are talking about, and you are assembling a self. So you are actually putting this protein alpha hemolysine on such a lipid bilayer, and you assume that it self assembles. So, there is a tendency of this protein to kind of go and embed into the lipid bilayer and kind of self assemble itself. What is also very interesting and important here is that if you look at the alpha hemolysine as a protein molecule, it really has a pore within it, it has a small narrow channel, and this also acts as a biological protein based pore. There are a lot of charge transfer processes across particularly our central neural system, nervous system, where we use this charge intake and charge outside delivery in order to transmit between the cells. Okay. And these are called exocytosis or endocytosis based processes, where there are essentially proteins on the cell wall which would open and close and give way to the charges. So the pore size illustrated here on this alpha hemolysine is about 2.6 nanometers. Okay. And as I have indicated before, one helical turn of a DNA is of about 2 to 3 nanometers in size, and so it very well matches with the 2.6 nanometer pore size on the alpha hemolysine protein. So, as a result of which, if you are assembling this kind of a structure across a cis and a trans chamber just in the manner that you have done for a cell, but in this case you make one side rich in DNA concentration, another side lean, and then apply an electric field, you should see the similar kind of effect as a drop or a dip in the current versus T plot to detect a single molecule, okay, a single DNA molecule if it passes by. So that is exactly what this principle is here. The recording that was made using this kind of a technology showed that you have these small peaks, dips which are coming out just because of the blockage of ion current as the DNA goes and blocks one of the pores. What is also very important for me to tell you here is that this really is a size effect. Okay, so what is also very important for me to tell you here is that it's really a size effect. That means if you have, let's say, a molecule poly A as opposed to poly C, and C as if you look into the molecular structure of C cytosine, it is a little bigger, it has 2 6 carbon 5 carbon rings essentially, and adenine on the other hand is a little bit smaller molecule. But if you can see here there is really a marked difference in the current values. In case of poly A, the current is slightly higher than in case of poly C, which means that if the size is more, let's say this DNA here which is moving through this porous membrane has a more size, it takes a while for the DNA to come out. So the average current which would be able to get generated in the case of a loosely fitted molecule is slightly higher in comparison to the current which you can generate by a kind of tightly fitted molecule in the pore. And also what is more interesting is that it takes a little more time for the DNA to pass through the spore once it is tightly fitted as opposed to the loosely fitted. Now this in a sense is kind of breakthrough. Okay, it gives us a feeling that someday maybe with this kind of strategy we could be able to sequence the DNA in terms of what bases it has, just based on the response of the current with time. And this is really one of the holy grails in the industry, how to electronically be able to sequence. And there are in fact companies now which are doing these electronic based sequencing, and at least there are a few products which are tried to be made commercial very soon about electronic sequencing. There are a lot of novel ideas and a lot of projects which are in this direction of how to quickly electronically detect the sequence, and people dream in the range of something like a nanosecond a base pair kind of readout of these kind of processes. Okay. So in this particular work though, that both RNA and DNA were tried, and they were observed traversing through the nanochannel structure, right, and that kind of gave a new direction to this whole area of manufacturing nanoscale pores. So one problem though about this particular work is that lipid bilayer, being a very fragile kind of system which can be affected by temperature, pH, a lot of these parameters, people started thinking of, okay, can we really produce synthetic pores made up of materials which are maybe thin wafers or polymeric materials. So there are a few groups in the world which have developed some of these synthetic nanopores, and one of the strategies that people have normally followed is using a SOI, a silicon on insulator wafer, as I would be just illustrating in this particular example. So here as you see, there is an oxide layer of about 400 angstroms, there is a silicon layer on the top of it of about 2500 angstroms, and this whole is assembled onto a handle layer which is a much thicker wafer, a couple of microns, probably couple of hundred microns, and this although it is not to scale, so it is a 4-inch SOI wafer with this handle layer. The thickness of the handle layer is about 525 micrometers, the SOI is about 250 nanometers, the buried oxide is about 400 nanometers. Okay. So what you do essentially is that you first grow thermal oxide on the wafer surface. Here you can see on these two sides, essentially you are growing the thermal oxide is about 1000 angstroms, about 10 nanometers thick, okay, and I'm sorry, 100 nanometers thick, and this you do on all sides of the wafer. So therefore both surfaces having essentially this thermal oxide. So once you grow the thermal oxide on the wafer surface, you open an etch window somewhere in one of the sides, as you can see in this particular illustration here. This etch window has been opened in the oxide, where you essentially make silicon as the selective layer and silicon dioxide as the etching layer. You design the etchant in that kind of a manner and use photolithography process to carve a small window of sacrificial resist and create a via through which you could actually etch away the oxide and create a mask, a hard mask of the oxide on the surface of the silicon. Okay. Once that is done, you can etch through the handle layer, just as illustrated in this particular example, using an anisotropic etch process. So essentially it is at an angle of about 54.34 degrees. Anisotropic etch, as I will be talking about in details later on, is an etch system where you use either some kind of an hydroxide to perform an electron exchange process as the layer etches away. So you are essentially converting silicon into the corresponding silicon hydroxide and dissolving it away into the solution. However, what is important for me to mention is that at certain planes, at certain crystal planes on the silicon surface, the electron release is essentially limiting the rate of formulation of hydroxide, because of the higher density of atoms there is a more binding of electrons, and so at a plane which is also called 111 along that direction, the etch step is the slowest. Eventually whenever you do this etching as an averaging effect, it kind of shapes in the shape of a 111 plane, which is at an angle of about 54.34 degrees with respect to the vertical. So that is how you get this kind of a window here. Okay. So you have to use an etchant combination, one which is selective to SiO2 and one which is selective. So it cannot etch this oxide once it hits the oxide, however it etches the silicon. So it is etching silicon and selective to oxide, and in the other system it is etching the silicon dioxide and selective to silicon. So you have to follow process 2 first and then process 1, so you can hit the right shape over this handle layer. Okay. So the etch top is the buried oxide layer in that particular case, alright, as you are seeing here. So then what you do is you actually take this upside down, and on the, so now you have an illustration here where you are talking about again doing a etch window from the top side. Okay. So you are actually, and I am not showing the bottom side here, the bottom side is actually shown here in this case. So you take this particular top portion as you are illustrated by this circle, and then you are doing an etch from the top side. So essentially this is that oxide 1000 Armstrongs layer which you had grown earlier on the top of this silicon layer, which is 1400 Armstrongs essentially, and here this is thermal oxide. So you can say that earlier the silicon was 2500 Armstrongs, but because oxidation is a diffusion driven process, the oxide layer formulated is about 1000 Armstrongs, making the silicon, the pure silicon, about 1400 Armstrongs or so. Okay. So now what you do is essentially you open another etch window here as you are seeing in this portion, and here essentially you are using again a photolithographic masking step and a resist which is selective to silicon but can etch away oxide very easily. So you can open a 230 to 240 nanometers etch window and then again perform another anisotropic etch. There would be some undercut in this case as well, so that this whole area here can be removed from the 1400 Armstrongs silicon layer, and you can design the thickness here in a manner. So because this is 1400, you have through calculations you can show that the lower part here is only reduced to about 70 to 80 nanometers. It is a very small part that kind of remains at the very end of this pit. So you have a case where you have opened an etch window from the back side and then use the same process step to open an etch window from the front side as well, and effectively you are just left with this 4000 Armstrongs oxide layer in between, the one which you saw here in between. Okay. So this is only about 400 nanometers thick, and then you have the advantage or the luxury of having a 70 to 80 nanometer hole on one side. So you can actually now use e-beam lithography and now etch or a hole which is about close to 30 to 40 nanometers in this particular region. You can also use the focused ion beam for creating such a pore. So you could actually design a system wherein you can actually open a 30 to 40 nanometer film, a small hole in this particular region here. Okay. In this particular 70 to 80 nanometer region, you can create a small hole which is about 30 to 40 nanometers. So once this is created, you put an oxide layer around it. Okay. So you again kind of grow, you first remove the buried oxide. So this essentially is the buried oxide layer, as you are seeing this, this 400 Armstrongs layer here is the buried oxide. So you removing the so the buried oxide almost in totality, all right, except a portion which is about 30 to 40 nanometers, and then you essentially regrow the 100 nanometer thermal oxide in this particular region. You are regrowing the oxide. So from this step to this step, after you have opened a small hole here through a FIB about 70 to 80 nanometers, you are essentially trying to remove this layer altogether and also try to grow so that you can shrink this pore from the general about 70 to 80 nanometers all the way to about 30 to 40 nanometers. Okay. And once this 30 to 40 nanometer is created by etching away this oxide from the bottom, you can actually kind of regrow the thermal oxide on the sides here. Okay. So, if you see here, this blue layer in this particular region, which I am now marking red, is the regrown oxide. So, you have step 1 where you are removing a small etch window on the back side, you are removing an etch window and etching on the front side, so that you are reduced to 70 to 80 nanometers pore. You cut using an e-beam lithography, a kind of this 230 to 400 240 nanometers width here of this particular oxide, and then you with an FIB kind of try to drill away this area, or alternately you can also remove the oxide altogether and grow or regrow about 100 nanometers of thermal oxide around this area, so that you are left with about 30 to 40 nanometers. Okay. One interesting point here to be noted is that if you heat this region using a focus beam, let us say you are using an e-beam, an electron beam, to kind of heat this particular area, you see that this oxide kind of reflows and tries to have a self closing gap. So, this 30 to 40 nanometers can be reduced to about 3 to 5 nanometers, as you can see here. Okay. So, you are regrowing the oxide and you are trying to flow it so that it kind of becomes self closing. This is illustrated here in the example. If you can see from this particular work reported by Decker et al. And later on Bashir et al. What they are showing here is that this is an excellent visualization process. So, you take a TM and try to start visualizing the pore when it is about 300,000 x. You can see this 41 into 26 nanometer pore, and then mind you, the magnification here is about 1,000 x, which is about 3 times this particular value, and here you are trying to investigate a 23 nanometer pore. And then if you look at the pore with time, as you are focusing the electron beam, the oxide reflows and the pore kind of starts becoming smaller and smaller. So, it starts self closing till it attains about 4.2 nanometers times 4.6 nanometers, and you essentially stop the visualization process in this particular instance. So, you are taking the bigger hole here, which is regrown with an oxide on the surface, and trying to focus the e-beam and causing self heating, so that there is a flow of this oxide and self closure of the pore. You could see the smallest pore size here, which is about 4.2 nanometers times 4.6 nanometers, and this is how size of a DNA would be, or it rhymes very well with the size of a DNA. So, you can make a synthetic nanopore in this manner, which you could later on use for DNA translation, and also try to understand or gauge whether it can do some sequencing. Okay. So, this is how these nanopore channel sensors are characterized, particularly for double stranded DNA structure. So, you can see that there is a micron size pore or there is a nanometer size pore here, which is in length about 50 to 60 nanometers, and in diameter is about close to 4 to 5 nanometers, and the DNA kind of is translated from the cis side to the trans side, and so essentially you can think of this side to be the positive bias here, this side to be the negative bias here, and the DNA translating across the spore. What is very interesting to note here is that you get upward peaks like this. Okay. So, the peaks here essentially are in the reverse direction. They are as if there is a large ion current suddenly being released after the DNA blocks and is about to cross over. So, this paper reported by Bashir et al., which I just illustrated earlier here in this particular example, it tries to kind of explore this principle, that why is it that we are getting a peaking action rather than a dip in the current level. And so, one explanation which is possible is given by this tendency of the DNA to kind of carry a counter ion cloud around it. We have been discussing off and on in the earlier lectures that DNA is essentially having a very strong charge on the backbone, a negative charge on the backbone. So, if it flows in a solution and it is essentially electrophoresing in a solution, there is a tendency of this DNA to grab together a counter ion cloud around it, and this shields the overall charge. As we have modeled before in electrophoresis, it kind of leads to a reduction in the charge by delta Z, and also increase in the radius, the overall radius, because it is moving along with the whole ion cloud in the center. Now the idea here is that as the DNA travels into this region and it kind of grabs together this ion current along with it, and as it goes out into the other region, it not only kind of introduces, or it not only kind of introduces a negative charge in this region, but also the positive counter ion group which it is carrying along with it. That is the reason for sudden spurt of increase in the charge in the ion pore. So you can think of it that as this is a positive ion, it kind of drags along the positive ion and it leaves into this side of the solution, and the positive ion starts coming back once it crosses through the pore, okay, and it comes back in this direction as the DNA is moving in this direction, and these whole positive ions here which come back or are pumped back into the cis side, okay, from where the DNA has translated into the trans side essentially. Because of this, there is a huge increase in current rather than reduction in current. So this essentially is an opposite effect than the micro scale Coulter counter, in which you have no such effect, because you are talking about a micron scale object. Whatever the flow is across that micron scale gap is a bulk flow, but here, because we talk about more in terms of almost to a single ion level what is going on, this counter ion effect becomes very important, okay. So you can think of that this counter ion cloud which is going with the DNA into the pore, when the DNA escapes, it is kind of released back into the solution in the reverse side, as a result of which there is an increase or a peaking effect rather than a dip down as is normally observed in other cases, okay. This is more so also because the silicon surface here essentially develops a charged dual layer, because you are talking about the same range as that of the dual layer between the silicon and the translating molecule. So this process is a lot more complicated than the normal micro scale Coulter counter. So essentially you can conclude by saying that in this case the DNA induces extra potassium ions when passing through the nanochannels. The interface current of the potassium ions does increase suddenly, and at the same time bulk currents decrease because of the DNA blocking, okay. So the overall effect is an increase in the current because of a sudden spurt of release of these ions back into the solution. So particularly in this example, a 200 base pair DNA has been used with a concentration of about 0.3 milligram per mil, and the electrodes used for this measurement are these AgCl based electrodes. As you know, this AgCl itself is non-dissolvable in an aqueous solution. So you have a coating of AgCl on the top of Ag. That is the way you use these kind of electrodes. We have talked in details in the electrochemistry section of this course. The bias voltage used in this case is about 200 millivolts, and the time of sampling is about 100 microseconds, which you would also get using a high resolution DAC card, a data acquisition card, okay. So this in a sense is actually, it has become a very major research area nowadays in biomems as to how you could actually measure the single molecular translation across these nanopores, and is it of some utility or effect in the diagnostics area. So there are other forms of sensing or detection which can be used with these MEMS kind of architectures, and so we had been talking earlier about silicon micro devices. So this is a direct application of a silicon micro device in biosensing, and this talks about mechanical detection or a mass based detection, which was kind of one of the frontier areas of sensing these days, okay. So here what this process is all about is defined by change in surface stress across a small thin film micro-cantilever. So you can see here this is a micro-cantilever which is coming all the way into the small pool, okay, and it is separated from the base, okay. So it's like a cantilever which is pointed into a gap here like a swimming pool. So it's a diving board and a swimming pool kind of arrangement, and here if you do something in the surface of this cantilever in a manner that the surface energy has a differential on the top and the bottom surface, you put some molecules you change the surface energy somehow by binding it to let us say some hydroxyl groups or some other SAMS layer, self assembled monolayer, in a manner that there is a differential between the bottom surface of the cantilever and the top surface of the cantilever, okay, in this manner. So you have something immobilized onto this region here on the top, so that you have a difference between the bottom and the top surfaces of the cantilever, okay. So there is a bending because of this, there is a mechanical bending, and this bending is also defined by something called the Stoney's equation, as given in this particular illustration here. So the bending delta z in this case, and delta z is this particular distance that the cantilever moves, is also directly proportional to a difference in the surface energies on the top and the bottom surface of the cantilever. So delta sigma 1 and delta sigma 2 here are the difference in surface energies on the top and bottom surface which occur because of this chemical change or chemical activation of the surface, right. And so therefore delta z here, the change in the z motion of the particular cantilever, is also directly proportional to the difference delta sigma 1 minus delta sigma 2, okay. So delta z again is the deflection of the free end of the cantilever, L is the overall length of the cantilever, T is the thickness, E is the Young's modulus, and U is the Poisson's ratio, and delta sigma 1 and 2 are change in surface stresses on the top and bottom surface. So this essentially is a mechanism of detecting if really there is a change. You can produce a yes no type of answer of whether there is a surface change, and I will just in a minute show another slide which talks about how you could detect DNA or recognize DNA of different base pairs, okay, based on this technique by just immobilizing some capture probes on the top of such cantilevers, such micro cantilevers. And really these cantilevers could be of all different kind of shapes. This for instance is more like a V shaped cantilever, as you can see, you know, this is actually a straight I shaped cantilever. So some typical dimensions of these cantilevers are illustrated here. It's about 200 nanometers thick film over which you make these cantilevers, and they can project as long as about 100 microns, okay. So it's really a very, it has to be a sturdy film, a sturdy structure which can go up to 100 microns and still be integral, although the thickness is only as low as about 200 nanometers. So this is what the aspect ratio is, about 5 times, okay. And they are normally made in silicon because the process is very well acknowledged and found out. However, there are initiatives to also make cantilevers in other alternate polymeric material with certain changes in their strength properties, modification in the strength properties. So let us see what we can do with this, and this is really an example, a very fine example bought from IBM's research plan on biosensing. So this is also corresponding to a paper reported by Fritz et al. In Science, which talks about how such instruments or techniques can be used for biodetection. Okay, so here in this illustration, as you see, there are two different kind of capture probes which are put on both sides of on a series of two cantilevers. So there is cantilever 1 and cantilever 2 here, on which there are these blue probes which are fitted on one side and red probes which are fitted on another side. So there are two different capture sequences altogether, right. And what we do is that in the first instance there is a green target here. You can see this green target which is corresponding or is just complementary to the red target which was already immobilized onto this particular layer of the cantilever, okay. So and this cantilever is also coated with the piezo material. So you have a kind of transduction of electrical into mechanical into electrical signal across this surface of the cantilever. So now when you flow this green probe, which is only specifically able to bind to the red probe, leaving the blue probe, there is always a surface reaction at this cantilever end here, okay. As you are seeing, the DNA is binding here, the red probe which was there on the cantilever is binding to the green target which is flowing by, whereas in the other cantilever here there is no binding, there is no significant binding, because this blue probe here does not really bind, it is not complementary with the green probe. So what would happen is that there is a change in the signal generated, and if this were to be the reference signal and there was a reference 0 in this area, there would certainly be differential which is being introduced here. So this is the differential signal that is reported with the time in minutes as the binding takes place. So as you see here, the binding is kind of ensuring that there is a pickup in the differential signal, and then there is a continuity here until now what we do is we flow another probe of similar nature, but now this time this is complementary to the blue probe here. Okay. So there is another target which we are flowing here, which is complementary, and it has the same number of base pairs on both the size cases to the blue probe here. So now the blue binds to this yellow target, and the yellow does not bind to all of these bound DNA only, and therefore this thing also bends, this cantilever now also bends. So this corresponds to a change back of the signal all up to 0. So the differential signal which arose here because of a binding of the green target with the red probe kind of changes back to 0 because of the binding of the yellow target with the blue probe. Okay. So this is how you could really make fast DNA detection possible by looking at just bending mechanisms associated with a pair of different cantilevers. Okay. This is an illustration of how these cantilevers would typically look like. You have all different shapes and sizes for these cantilevers, as you can see here, and this was actually reported as a module by IBM in its Zurich Research Center for the first time, where they talked about how DNA hybridization can be carried out using mechanical signals. So this was kind of one of the first initiatives in the area of how sensing can be in terms of transduction into mechanical signals from chemical signals. Okay. So this in essence is some of the kind of range of work that has been covered in the area of DNA related detection and sensing using biomems kind of architectures. Alright. Okay, so this kind of brings us to the end of this particular lecture. So in the next illustration, the next lecture, we would cover topics or different topics or aspects related to proteins, and then we will look at how the living cell normally correlates between the sequences of the RNA and the proteins, and we will find out more, as in fact a very interesting topic of the process called transcription and translation within a cell. Okay. Thank you.